Welcome to Neurosalience, the OHBM podcast.
Hello and welcome to OHBM Neurosalience podcast. I am Omer Faruk Gulban, the current podcast team lead of Organization for Human Brain Mapping Communications Committee. In this episode of Neurosalience, we are going to have a conversation with Peter Bandettini, and we are going to first talk about the previous season of Neurosalience. Peter will give us some of his highlights, and then we are going to talk about the upcoming episodes in the current season, season 5. In the later half of the discussion, we have talked about some of the current topics that Peter was interested in, such as resting state fMRI and deep sampling of individuals to modeling of the brain and different approaches there. And finally, some thoughts on the evolving publication scene in neuroscience. I hope you enjoy our conversation and the upcoming season. So, hello, Peter. Welcome.
Thank
How are
you.
you doing?
Doing fine.
Could you rest it well in summer? We had a very busy season last year.
Yeah, it was busy, and but I keep on, I keep on doing this, and I'm really enjoying doing this, and I always tell my guests that I benefit a lot. I learn a lot as I sort of research the guests, and it sort of helps me keep on top of the field for myself, but also hopefully it helps everyone else as well. Just bringing out perspectives that are a little bit different than just talks or just reading their papers or just talking to them at conferences. This is sort of like attempting to sort of get more into like some of the issues with them and maybe look at their personal side and look at the wider context of the field and how they think about that as well. So it's fun. It's been fun.
Yes, it's great to hear, and we are happy to have you still as our host. I have some questions for you. Maybe we can switch to those. So
Sure.
I wanted to ask the following first about the previous season. What has been the most memorable podcast episode or interview in the last season? You can also, it doesn't need to be only one, but maybe you can talk about multiple as well.
Yeah. I mean, it was a whirlwind of people, and I thought about that question. I mean, you sent me the questions before, but I thought about it, and I thought, well, not any one stood out completely. There's some that actually I feel like go better than others, just because I feel like I'm, maybe I've had more sleep that night or whatever. So I'm just gonna maybe mention some of them, and talk about what I found most interesting. So it's the season started off with Russ Poldrock, one of the first guests, and I'm always impressed with how prodigious his productivity and how clear his thinking is in terms of setting up these tools and setting up this environment for open science. And he has many tools, and he's really fostering and catalyzing the field in that way. Another person sort of devoting his career to educating people is Andrew John, who we had on. He's a person who's known for education in neuroscience. He won the OHBM Education Award, didn't he? Yeah. So he actually creates all these resources that I think it's really worthwhile to take a look at this. And it's profound what he's been doing and what he continues to do. It's super helpful, especially the new students and new professors or anyone. I also talked to Alex Huth, who, as most people know, worked closely with Jack Gallant, but he's since then differentiated himself from Jack's work, but also added his own rigor. He's now continuing his voxel-based predictive modeling, but then going into natural language models and things like that, which I really think is really powerful and really unique. Shella Kilotz was a great guest. She's always, we've had her on now twice. I mean, she was talking about the imaging neuroscience migration, but also this time we talked about her own work. And she's mostly known, I think, for her work on characterizing quasi-periodic patterns in resting state. And I think she's delving into resting state deeper than most people are. And I was really also impressed as I talked to her in doing my research. She has this nice method. It's sort of an iterative seed-based approach that sort of pulls out these slow fluctuations, which which I thought was worth looking into and really good. And she's actually doing all the really hard work of trying to correlate it with EEG and other things like that. So I think it's definitely work to be continued to be followed. So Evan Gordon, I think this was one of my favorite podcasts because, everyone knows about deep sampling, trying to image individual subjects. And he was part of the Midnight Scan Club at WashU. And Evan Gordon has been really doing innovative stuff. I mean, obviously he had a popular paper that came out almost a year and a half ago now on finding an area in the motor cortex that sort of is the hub of other areas that hasn't previously been found. I mean, motor cortex, we all thought it was all figured out for the most part at our scale, but no. And he found this by doing deep sampling, taking the Midnight Scan Club, having them being scanned for like 5 hours. And I was actually, the thing that surprised me most was my own reaction to this. I came away from that podcast thinking, this is the way that things should be done. This is how fMRI will advance. And I haven't had that feeling of, I generally am enthusiastic about everyone's approaches, but for fMRI to truly advance, I think we have to get more data sets of there's much more information you can get using 5 hours of data as opposed to 1 hour. There's so much more there. So I think that just blew my mind. And obviously everyone knows this, but I actually have to just put an emphasis on this, that I think it's really an important way to go. Monica Rosenberg is a new professor, sort of starting out, forging her way in Chicago. She's continuing on her theme of looking at fluctuations in attention, among other things, in time, which I think is actually another really nice individual subject use of fMRI. Marcel Meselam, also in Chicago, but there towards the end of his career. One message that I got from him, aside from everyone should read his classic paper, From Sensation to Cognition. And that paper is not only prescient, it was way ahead of its time, but also unbelievably good writing. It was just one of those things that I was more distracted by how good the writing was than the content. The content was, of course, amazing, but very fun paper to read. Nathan Spreng. Nathan was a poet. He still is a poet. That was a direction he wanted to go for a while. So that was actually really interesting to know. Jack Wells is doing pioneering work. Among the MR physicists who care about fMRI, it's kind of like one of the holy grails is trying to understand or try to find a way to image the glymphatic system of the brain. And Jack is pioneering that. And so that was a great episode. Gang Cheng. Gang is a friend of mine. He's a colleague. His office is right down the hall from mine. One other little known fact is that Gang and I are exactly the same age. But aside from that, Gang is great because he's one of the only statisticians in the field, but he's not just finding better ways to get a better p-value or a more correct p-value, which I think I've always appreciated that, but I think it's actually the wrong direction for statisticians. He's the one saying, wait a second, we're throwing out all this information. We have to look a little bit better at our data to not throw out all this useful information. Let's take away our thresholds. Let's look at every single nuance and fluctuation because that contains information. He really has a great perspective on pulling out information in a very rigorous way. Danielle Marinazzo, he's done many things, but one thing actually that struck me that opened up my mind to his work is the progress that's been made in characterizing the hemodynamic response in resting state on a voxel-wise basis. Every single time we do resting state correlation, there's this implicit assumption that all the signals are in phase and they're all shaped the same way. And so a lower correlation means lower connectivity. What he's been trying to do is say, hey, wait a second. No, we have to actually calibrate the whole brain. And he has some various methods for doing it that are good. I don't think they're perfect, but they're good. And I think it's a thing that all of us need to think about. When we make our correlation maps and we say it's connectivity, there's many, many things unrelated to connectivity that are due to hemodynamics that we need to account for to really be able to say, and even then, it's hard to really make that leap to saying it's connectivity. Rotem Botvinnik-Nezer. She's also a young scientist now. She's at Dartmouth. She wrote the classic paper, which is co-authored by many people, on the many data sets analyzed different ways. And one thing that I was pleased that came out from that discussion was how measured she was in terms of, some of the concerns I had with that is that it was sort of setting up a straw man. It was setting a threshold high and actually emphasizing how all the analyses are different. And then obviously popular press picked up on this and they said, hey, everything's, fMRI gives different results when you analyze it different ways. Well, that's kind of true, and she sort of came out with the idea, she agreed though that, hey, if you lower the threshold a little bit and you broaden your perspective, they're actually really similar. So you have to actually step back and say, hey, it's important to actually, and this sort of ties back into maybe Gang Cheng's podcast, that yes, the paper emphasized one point, but the broader point is that maybe if you did lower your threshold or set up your questions a little bit differently, people look more similar than different. The results are more similar than different. Peter Fox, I've known for years and years. He was one of the first people to be a promoter of data sharing with his brain map database before even OHBM existed. He was promoting it. Let's pool our data. He was mostly doing center of mass coordinates. And the other thing is, it emphasized to me something kind of close that I follow closely is this question of what is the physiologic mechanism behind the bold response? Everyone knows oxygenation goes up, but in my mind, it seems like over the years, it's become settled. Like, oh, that's to deliver more oxygen to the area that's active. Peter's been saying that, in fact, glucose metabolism goes up, and it's much more than CRO2. And he's done experiments to suggest that brain doesn't need this influx of flow to function as far as burn oxygen. And there's recent papers coming out that sort of suggest that maybe the increase in flow is to maintain a steady pH and to get rid of CO2. It's a CO2 driven effect and it's to get rid of the CO2. So, basically to clear out the waste from metabolism. And that, to me, makes a lot of sense. And so that was kind of opened up this chapter in fMRI that everyone thought was closed, but we're still trying to figure out why we get this amazing robust bold effect in the first place. What's its purpose? So that's ongoing.
Actually, after that episode, Peter, I went into reading a couple of papers, and I encountered a paper called Neurovascular Coupling, Motive Unknown by Patrick Drew.
Yeah.
And I had a feeling that he would be a good guest as well, because he was there in that paper, I think, arguing for 5 different explanations.
That's great. Okay.
I think I never mentioned to you, I just remembered now.
Oh, that's great. That's great. No, that's actually good to know. All right. Now all the listeners can see our process here. So we can, Patrick, if you're out there, I'll be emailing you. It's kind of esoteric to think about in the context of brain mapping. Because it works and no one really cares. I mean, we care, but at the same time, I think what's cool about this is that new physiologic information that might be clinically relevant might be derived from better understanding this. And that's sort of the goal, to sort of make fMRI more clinically relevant. Maybe it's not in finding biomarkers for neuropsychiatric disorders. It might be much more for understanding neurovascular coupling to predict Alzheimer's or something like that. So that's where the interest is. So Vince Calhoun was my last guest before we started doing the episodes sort of tailored to OHBM. And I had him on twice. Once, half the episode, I thought it was, and I asked him if he was comfortable talking about this. I mean, I've known now in the last few years, I've known several colleagues who passed away. And to be honest, Vince came pretty close to it. He had cancer and he beat it. He was lucky to beat it. And he talked about that a little bit in the episode. I personally think that it kind of changed him. To be honest, I think that after that, it was around 2014. After that, he's been on a tear, and he's been by far the most published author in brain mapping, and he's just been on a mission, and he's doing incredible work. And so I think it gives one perspective, definitely. He's also obviously been creating amazing tools. He produces wonderful tools for looking at dynamic connectivity and things like that. But he's now focusing more on multimodal fusion. And I think that it's worth looking at his websites and looking at his tools for those of us who are trying to fuse EEG data with fMRI data and so on, he's ahead of the curve. And of course, Todd Woodward also was interviewed. He actually has a really unique approach for pulling out fMRI information. He focuses on schizophrenia. It was wonderful talking with him. I think it's worth reading some of his papers because he's sort of forging his own path, kind of separate from the mainstream in terms of analysis. And yeah, it's worth considering what he's doing and looking carefully at it. Yeah, so some general observations. This is what I get from talking to everyone, and even from the past other for season 1 through 4, is that the people I talk to are, not only, they're really prominent, and some are less prominent, but they're all really smart. They're all highly motivated, skilled. But I think more importantly, they have a kind of a rebellious streak. A lot of them, I think that when we're trying to create new knowledge, that's what science is pretty much all about. We're trying to create new knowledge and new insight. You always have to be counter to the standard. You have to sort of have a mindset of wanting to find something that's different and deeper or more insightful. And I think that they've figured this out. They've had it in them from the very beginnings of their career, and I think that that's actually comes across really closely. Like, for instance, Marcel would he would create like a bubble in terms of buffering everyone from the administration and just kind of doing their own thing in their own way. A lot of people do that, and I think a lot of people that I find, like, I find a growing trend potentially, and maybe it's always been a trend for younger scientists, that a lot of them they're kind of afraid in some sense. They want to do work, and they want to make sure that it agrees with the previous work, and they just want to say what they did and make sure it's correct, which is true. You have to do that, but it's more important to convey the insight of your work and convey how it's different than the previous work and how it builds on it, and how it's I think one should try to do that as opposed to trying to fit in.
I see.
And all these professors, all these people I interviewed do exactly that. They stand out, and those are the ones that have the most impact in the field.
Thanks for the recap, Peter. So now I would like to switch to the upcoming season. What awaits us in the upcoming Neurosalience season?
Yeah. So first of all, I got the season off a little bit delayed just because my own schedule was just crazy, and I'm trying to fit this in wherever I can, and I was able to finally fit it in. So the people so far, so we already recorded an episode with Angie Laird. Her episode, I think it will be one of the first coming out.
It will be basically the second episode after this one. Yeah.
Yep. Okay. I just last week had a great discussion with Ellen Evans, who's at the Montreal Neurologic Institute. They've also created a lot of amazing tools, and they're all about big data. And Alan's been a pioneer from the very beginning. Was one of the founders of OHBM. So that was a great discussion. In a little while, in next week or this week, I'll be both Faruk and I read this book. Faruk mentioned the book to me, and I read it, and I liked it. It's called The Idea of the Brain by Matthew Cobb, and Faruk and I thought that he would be great to have as a guest, and so we're going to pick his brain about that book and other things. Kind of it's the idea of the brain. It's basically looking at what we thought of the brain over time. It's historical, but it's also kind of a reflection on the field and how they thought of the brain, as opposed to sort of building what the brain is. It's like what we thought of the brain and sort of putting our own perspective on that. So that was great. So I'll be talking with him this week. I was lucky to get the agreement of, with the help of Ravi Menon, I got Seiji Ogawa, the only person who I think is in the running to actually getting a Nobel Prize.
I didn't know that.
Well, I don't know. I mean, I say that, but and people say it probably jinxes him. But I think they're waiting on fMRI to have more of an impact.
Oh, we need to be working faster then.
But I think Seiji, as all of us know, is sort of a legendary name, and he was the first developer of bold contrast before fMRI existed, or developer, discover, whatever. He studied. He actually, his main area of study was red blood cells, and when he brought this out. I mean, the understanding in the field was that, susceptibility changes in blood aren't big enough to show anything, and nobody actually tried it. And so he did it, and it worked. Obviously, just as a personal anecdote, I've known him for years. He was on my thesis committee when I defended my PhD. So we have a close connection in that regard. And he's 90 years old. He's still very active. Ravi mentions he's now learning MATLAB. He's retired, but he's still on top of things. Yeah, no, I'm looking forward to talking with him. Other people that have agreed so far to Max Shine, University of Sydney, Sepeda Sagdiani, who was also on our panel. I wanted to focus in on some of the research of the people on the panel at OHBM and for the live podcast. Yeah. Simon Eickhoff is going to be on, and Michael Millam has agreed. We haven't figured out a time yet, but he's agreed. Another researcher who caught my attention when I was visiting Max Planck, Charlotte Gross-Weissman. She actually has some really cool work. She studies child development and looking at development of white matter tracks and how it corresponds with behavioral development in recognizing faces and things like that. And so it was really cool. And she's a younger scientist, and so that gives a perspective of what the issues they're dealing with.
Yeah.
So that's the next season so far. I'm still inviting another 10 or so people, so we'll see what happens.
Okay, exciting. Also, things change sometimes when we are editing the episodes, or sometimes a paper comes out and we want to quickly shoot an episode. So let's see, but it's already exciting. I'm looking forward.
Yeah, I'm looking forward to it too. Yeah, and I was actually thinking, I think one of the shifts in how I might do some episodes is, less about, certainly the people themselves are interesting and worth talking to in terms of their career, but maybe having people on in relationship to bigger issues in the field that we're all sort of trying to think.
I see, like more kind of topic focused, or
yeah,
like a discussion, debate kind of focused maybe episodes. Yeah.
Yes. Yeah. Because there's many, and maybe just to shift into it. I mean, there's all kinds of issues that are near and dear to my heart that I want to bring out, like things like all the efforts for modeling the brain. How are there's so many different models, and how are we going to bring them all together? Obviously cutting edge fMRI methods. That's my wheelhouse. I like to talk about it. Mechanisms of brain function. How are we going to get at from mapping to mechanisms? Things like multimodal integration. How is AI having an impact on neuroimaging? Obviously, clinical inroads for neuroimaging, that's one that's still a challenge, and it's an important one. And everything from finding biomarkers to the nuts and bolts of actually implementing it clinically, which is tough. Publishing in neuroscience is a big topic. We'll probably talk about that a little bit more. The question of what really is resting state in resting state fMRI. People are honing in on things, but I think it's unknown. Single subject imaging doesn't have to be deep sampling. And then as Faruk, as you mentioned, as papers come up, we'll try to highlight them if they're really hot papers. So
yeah, actually, Peter, now you mentioned it. Recently, I have had a participant that went into the scanner we were testing a set of new anatomical imaging protocols. And when she went out, she said that this has been the most active I felt when I went to any fMRI or like MRI study. And it made me think she was basically in the resting state the whole time for 1 hour, right? Like doing nothing, not even watching anything. And she was saying that like she felt the most active, even more than when she was invited for task fMRI experiments. But that kind of made me question, indeed, like what are some maybe misconceptions about resting state, or is it like really resting when we even like call it resting, So do you want to like talk a bit about it, or give your perspective
Sure.
or experience?
Yeah. No, actually, I thought about that. Yeah. Certainly, the brain is never at rest. I mean, that's and it's becoming more clear that there's this intrinsic ongoing activity that seems to reflect whatever activity your brain was active in hours before or days, or maybe up to days before at the most. And so why is that happening? We don't know exactly why. And I think that thoughts, like you're saying, like having active thoughts. One of my staff scientists in my group, Javier Gonzalez Castillo, is sort of actively pursuing this, sort of like trying to understand the spontaneous conscious thoughts in the scanner. And it's very hard to do that because it's sort of like the equivalence of Schrodinger's cat in quantum mechanics is that, if you ask them what their spontaneous thoughts are, suddenly they're doing a
I
task. And
see.
so it's hard to probe that information during the scan. You have to sort of do it afterwards, which is, I'm always skeptical about that. I mean, if you were asking me, it's like, I don't know I couldn't specify the thoughts I've had in the previous 5 minutes. I mean, you can kind of do it, but I think that spontaneous conscious thoughts are just the tip of the iceberg. I think that we never are able to predict what our next thought, our spontaneous thought is. And I think that's intrinsically related to all of resting state. All of resting state spontaneously happens, and so every once in a while, a network that we have conscious access to is stimulated or activated, and then we have that thought. So I think it's sort of the tip of the iceberg of all these processes going down on all the way down to subcortical level, things that are well outside our conscious awareness. And I think one of the big I was actually kind of surprised with this. I mean, I've recently I was just talking with a prominent, not a prominent, but a really active person in looking at resting state, and we had this debate. And he's like, well, resting state is 0.2 to 0.1 hertz or something like that, and when you put it in that framework, it implies like there's some oscillation going on, and everyone is thinking, oh, these are resting state oscillations. They're just a sine wave, which nothing could be further from the truth. Some work that am sort of overseeing with my postdoc Josh Faskowitz, who's sort of developed this edge analysis approach, kind of highlights the point that no, it's resting state is these punctate moments of activity that are separated by long periods of time, and overall it kind of looks like this low frequency oscillation, but it's actually points of activity that in some ways everyone kind of knows, but everyone isn't sort of connecting that with resting state analysis in some way. It's kind of interesting how there's this, people hold both ideas in their head, and it's a little bit fuzzy still.
Yeah, I think it also kind of goes back to the like I always think in the framework of this idea of the brain book by Matthew Cobb, the way we indulge ourselves with the current technology, like computers, electronic devices, like that they oscillate and they do stuff like in periodic terms. It kind of in a way forces us to think that brain should be working like that as well, but this can be a nice scaffolding to put your ideas around and impose like some sort of framework to understand how the brain works, but also it can misguide you in certain ways.
Yeah, and also, and along those lines, and it kind of came up with my talk with Ellen Evans, and I give a talk usually at the beginning of some of my talks. I talk about all the scales of the brain, all the temporal and spatial scales. I mean, the brain is organized in networks all the way down to individual cells. And, with fMRI, we're sort of biased towards looking at this macroscopic network sort of thing. And I think you're right. I think that our framework now is, for better or for worse, it's sort of like network based. And I think that's useful, but are those networks at that scale really going to be that informative? And I think they might be, but in fact, there was a really interesting paper I read recently that talked about an individual neuron having an ability to, basically, it's a prediction machine. It's in itself. It sort of it tries to predict and anticipate other inhibitory and excitatory inputs, and then it adapts accordingly to minimize its energy to adjust to those. So even so the network goes all the way down to individual neuron. And I think we have to reframe our perspective. And one thing, it goes way back to one of the first episodes when I was talking to Danny Bassett. And I asked her this because she does everything in networks. I mean, she's the network pioneer. And I said, well, wow, it seems like everything, we can actually think about the brain completely in networks, and it's really powerful. And she's like, and I said, is there any other way of thinking of things? And she's like, well, there's fluids, there's that aren't, you could think of it as a fluid. And I think that if you have a conglomeration of neurons that are forming these networks, on some level, it's sort of like a fluid that, you could almost, I mean, she probably didn't meant to imply that, but that's what I took from it, that we need to potentially, we might benefit from being flexible in not sticking to purely network-based analysis and sort of try to model it in terms of a fluid. I mean, obviously Michael Breakspear models it in terms of waves. That's sort of like a top-down sort of imposing these waves on it, which I think is useful, but also just looking at it from bottom up each neuron has its own environment in which it's trying to optimize, and sort of that's almost like a fluid sort of state. So
Actually, I think the fluid analogy is quite useful in many fields of science. I remember reading in a couple of different books actually, like they always go back to this like fluid dynamics equations and then try to derive stuff from there in various different contexts. So maybe we should have a fluid dynamics episode one day
yeah,
in Neurosalience.
I agree. And one other person that also sort of along those lines, which was really an interesting talk. Was Nicole Rust, who has written a couple of papers and just coming out, I think, with a book in about a year. And she actually kind of has the view that modeling the brain is kind of like trying to predict the weather. It's a chaotic dynamic system, and unless you absolutely know the initial conditions, your predictions will be way off very quickly. and if what we want to do is understand the brain such that we can predict behavior or even modulate behavior, can we change to the extent that if we had the tools for changing the weather, could we really? So that was actually a thought-provoking. She's given this talk. It's on YouTube, I think. There's various versions. Nicole Rust, and I would like to have her as a guest. I think
Yeah,
I'll invite her.
that sounds interesting.
Yeah, I don't know if it was meant to be a bleak, not really bleak, but it's sort of like, hey, the brain is more complex and has more surprises than we're aware of still. And
Also, now this makes me connect to a paper I read a few years ago. It was about like it was published in I think Science. It was like a kind of a big opinion piece, and there they were saying that there are just some regimes in predicting the weather. We know that it is chaotic by nature, so like by definition we cannot know what will happen. And then, but when you switch to the microscopic scale, then you can kind of I don't know, like look at the crystalline structures and how the ice forms and so forth. But in the macroscopic scale, you can maybe look at the clouds, how they form and what they would affect. But like there is this intermediate scale that is chaotic, and then they were talking about how to deal with it. Maybe like if we find the right guest, that could be fascinating to make these connections between like brain science and who knows, even like meteorology, you know?
yeah. No, that's actually, I think that's really interesting, and I think it's waiting for some good modeling people to try to do this. I mean, because now it's like, I think the hope is that, and this is a big part of Human Brain Project and other things, that if we understand all the circuitry really well, we can then go in and tweak the circuits, and then that will help people with schizophrenia or whatnot. I don't think it's that simple. You can't, sure, we might understand every single cell, we might understand every single interaction, but we understand, like you said, the details of how weather is formed. We understand that in principle, but we still can't control or predict it.
Actually, I should send you that paper
Yeah,
maybe.
that'd be great.
Today, for instance, I couldn't predict the weather 1 hour ahead. It was still raining. I was annoyed coming here. It's like, why is it not working? You know, like why they cannot tell me that it will stop at this hour?
Yeah, it's intrinsically. I think now the theoretical limit, based on our measurements, is like 7 days to just any reasonable degree of accuracy. But yeah, you almost have to have right. You almost have to have like a measurement device every square meter to be able to start to make more accurate. But still, it's really hard.
So this makes me wonder, Peter, about your comment on the Evan Gordon episode, and you said that, or maybe we should sample the individuals densely, right? It's kind of the similar parallel here. Like maybe to understand how the brain works better, we need to have dense sampling. So I am very interested in this because I work on ultra high field exclusively, and I like doing few number of subjects, many hours, even for anatomical purposes, like a couple of hours of different types of data. And I find it valuable to see all these details of the brain. And I'm thinking, what are the current limitations of doing this deep sampling? Is it like, I feel like it's mostly maybe publication bias, like, or people think that they will publish in a harder way, maybe, or is it like a funding? Do you have some thoughts about it?
Yeah. Well, I think actually, I mean, the way people have thought about it, I mean, it all started with the way back when people were doing PET and they needed this average across subjects and the resolution was kind of low to get any sort of sensitivity. And it sort of evolved in fMRI, sort of like, there's two types of problem. I mean, fMRI actually, even from the very beginning, we did individuals. I mean, the high impact vision neuroscience papers were usually 2 or 3 subjects who sat in the scanner and you mapped out their retinopathy or their, V1, V2, V3, so on. And you did it really well. And nobody actually questioned like, oh, well, that's just one subject. How do you know that's going to happen? And everyone that was sort of like, okay, this is something worthwhile and novel. And that sort of has gone away a little bit in other domains. And so I think now people are like thinking, okay, if you want to compare populations, you need to have a lot of subjects. And that's true. That's true. The Merrick paper sort of illustrated some of the problems with that, is that you have a lot of subjects. You need, and according to the Merrick paper, which was one of the best papers on so many different levels that I've read, pointed to the fact that, oh, wait a second, you might need 2,000 subjects to draw any inferences about populations. And so that was like, everyone's like, oh my gosh, what are we going to do? And I think that is forcing us now, I think, to take a few big steps backwards to say, wait a second, there's a lot of variation across individuals, even healthy individuals, that we can't necessarily pool people and assume that their disorders are homogeneous or their behaviors are homogeneous. And two, we can't pool their brains as easily as we thought. Like when you put a brain into a template, even if you just parcelate it with maybe 200 parcels, those parcels are very different for each subject. And so when you're doing this normalization, you're probably mixing a lot of the parcels. And so your differences might be due just to that. And so we have to take a step back to figure out better ways of parcellating, and then also better ways for understanding all these anatomic, functional, and behavioral differences in subjects. And so, and what they did was, I mean, one of their papers with Evan Gordon was, they used like maybe 12 subjects, I think. It was part of the Midnight Scan Club. They scanned them all deeply. And they were even able to find interesting differences between those 12 that were not previously appreciated. I think the field probably should spend more time scanning individual subjects at least 5 hours, because really, like you were saying, the quality of data that comes out of it, because from having data that's self-registered because it's the same subject, the detail in which you can actually, that detail is not just more detail, it's actually new information.
Mm-hmm.
And there's more there. I mean, Javier Gonzalez Castillo, again, he wrote this paper in 2012, average, having a subject just do a simple task, a simple visual motor task, averaging for 9 hours. And they found the entire brain, if you loosen up your analysis and just look for anything that's time locked, the entire brain is active or deactivated in some way. And that's all new information that we never appreciate as well. So I guess my long-winded answer to this is that I think that the field is best positioned to scan people deeply and then re-look more carefully at the precise differences between individuals, and then move forward slowly with trying to start to pool people and to compare. But how do we even, that's another big problem now, as we go to higher and higher resolution, like, with layer fMRI. How do you spatially normalize that data across subjects? It's nearly impossible. So you have to collapse in other dimensions, that are less variable. You have to do something else. So right now we're limited. We have to do individual subjects with layer fMRI because we can't do simple, like, put it into a template and average across subjects. There's no way. It's like a fingerprint. It's so many differences. So it's a real problem. And I think even studies that are looking at things correlated with behavior, you can find, I think that, there should be a lot more studies really scanning people deeply and then progressing slowly beyond that, and then regrouping and figuring out how we should treat, large N databases, data sets.
Also, on a related note, I sometimes feel that we are so spoiled because 100 years ago, when Brodmann, von Economo, Koskinas, and others, like, they were doing their neuroscience work, this was, I don't know, probably tens, if not hundreds of hours of getting brains, staining them, sectioning it, looking at it under a microscope. Like this was such a long, like like you need to spend a lot of time investment to get anything out of that sort of data. And this
Yeah.
was all individual brains. And now, I mean, sure, like our machines are very fancy, much more advanced technologically, but in a couple of hours we can get the whole brain scanned, and we can put anyone, they don't need to be dead brains inside. And I have a feeling that we have some duty there to do as well, and look into detail, and also like realize that, hey, like 5 hours per person is actually not so much compared to
Yeah.
the historical perspective of doing neuroscience 100 years ago. It's actually very little. It's super fast.
I totally agree with that. Yeah, I remember early days of fMRI, Steve Peterson at WashU would lament that it's too easy and there'll be too much just stuff. And I think that he was sort of thinking along those lines. And I just wanted to emphasize also one more point, that sometimes is the wrong message taken from the Merrick paper is that, oh, fMRI is not sensitive enough and we need 2,000 subjects. No, no, no. I think that fMRI is more than sensitive enough. You can put a single subject in the scanner, and even with one session, you can easily map out their entire resting state connectivity. Obviously, you do it better the more you average, but people are already using this to some degree. One of the best papers also from WashU, Nico Dozenbach, with the study with putting a person, their arm in a cast, and then finding that their resting state connectivity between left and right motor cortex altered within hours of putting the arm in a cast. And so that shows that with a single subject, with not that much scanning, you can actually have potentially clinically useful information. So the sensitivity is there. With a single event, you can actually see a response. So the sensitivity is there. It's just how we pool the data. Well, the sensitivity is there and it will grow. It's probably optimized around 5 or so hours. And then how we actually combine that data makes a difference. If we don't account for every subject difference, both anatomically, functionally, and behaviorally, then we're going to mess things up. So, yeah.
Okay, so now we can wrap it up here. I had another question about the evolving landscape of scientific publishing, but actually now I'm thinking we could even make a separate episode for it.
Yeah. I was actually, so that's one of my ongoing projects, is to find some people to invite on the podcast to actually have a discussion about the future of publishing. It's those in the field that aren't aware of this. So there's this lawsuit put out by Lucina Uden. So I can't really interview her when she's in the middle of the lawsuit because I guess you can't really talk about it. But basically suing all the publishers, all the big publishers, basically saying, look, you're charging these exorbitant fees. You're making these, big profits, which is not illegal in any sort of sense, but you're not paying the reviewers, which is interesting because, that's one angle on this. I think that, we're working for free, and it's sort of like, we're not only working for free, but then we're paying to submit our papers, and then other people, if they're don't have open access, they're paying to see the papers, and the NIH is like, or NIH or any funding agency is paying like twice or three times to the publishers in different ways. So that's sort of like the bigger picture, but the angle of paying the reviewers, I think is important. I personally don't think it's a good idea to necessarily pay reviewers. I think it's the wrong incentives, and it should be part of our job. But that's my feeling, and it could be not exactly correct or whatever. But I think that we'll have a good discussion. I'm going to try to invite a bunch of people, and we'll have a good discussion about that. So.
Yeah. I just want to know that ChatGPT is not reviewing our papers. Sometimes I'm not sure these days.
That's true. Yeah, that might be also worth a podcast. The effect of ChatGPT on papers and everything.
Thanks, Peter, for today and spending a bit of extra time so that we can kickstart our new season, and looking forward to the other episodes.
Okay, well, thank you. Looking forward to it.
Neurosalience is brought to you by Organization for Human Brain Mapping. This week's episode is produced by Omer Faruk Gulban.